I want you to picture something for a second. Imagine you are driving a car. But this isn't a normal car. This engine, your biological engine, has to run 24 hours a day, 7 days a week. It can never, ever turn off.
No.
If the engine stops for even a few minutes, the car is totaled, game over.
That is a very high-stakes commute.
Right. But here is the catch. You can only put gas in the tank maybe 2 or 3 times a day. And sometimes, you might go hours and hours, maybe even a day without passing a gas station.
Right.
So you have this massive mismatch. You have a constant, unrelenting demand for fuel, but a totally intermittent supply.
That is the fundamental paradox of being alive, isn't it?
Yeah.
If we didn't have a bridge between those two things, supply and demand, we wouldn't survive our first night of sleep.
Exactly. And that is what we are unpacking today. We aren't just talking about sugar storage. We are doing a deep dive into the biological savings account. We are talking about glycogen.
And it's so much more than just a savings account. As we went through the research for this, it became really clear that glycogen is, well, it's an insurance policy against chaos. It's a strategy for managing uncertainty.
That is a great way to put it. So, for today's deep dive, we have pulled 2 absolute heavyweights off the shelf to guide us. We've got Leninger Principles of Biochemistry, the big red book, which is going to give us the molecular nuts and bolts.
Think of Leninger as the engineering manual. It explains the thermodynamics, the structures, and the deep logic of why the molecules are built the way they are.
Exactly. And we are pairing that with Harper's Illustrated Biochemistry, which brings the medical reality. It tells us how this stuff works in a living, breathing human and, a little terrifyingly, what happens when the machine breaks.
And that contrast between those two sources is really where the magic happens today. We're trying to synthesize that structural logic with the clinical reality.
So before we jump in, just a quick disclaimer. We are going to go deep, but this is an interpretation and a synthesis. We want this to be a conceptual map for you. If you are a student or a clinician, please don't let this replace reading the texts.
No, absolutely not.
Use this conversation to make the reading actually stick.
Exactly. The goal here is to give you those aha moments so that when you look at those dense pathway diagrams in the book, they actually make sense. They're not just a bunch of arrows.
Okay, let's do it. Let's start where Leninger starts, with a basic, almost childish question. We eat carbs. We digest them into glucose. We want to save that glucose for later. Why don't we just keep the glucose? Why do we go through the massive energy expense of building this gigantic, complex, tree-like structure called glycogen just to break it down again 6 hours later?
It seems incredibly inefficient on the surface. You're building this complex castle just to smash it down.
Right. Why not just have a bag of glucose sitting in the cell?
Leninger answers this with a thought experiment that honestly blew my mind the first time I read it. It isn't about energy storage per se. It is about physics, specifically osmosis.
The water problem.
The water problem. Let's just, do the math for a second. Imagine a liver cell, a hepatocyte. If that cell tried to store its normal energy reserves as individual free glucose molecules, the concentration inside that cell would be about 0.4 molar.
Okay, 0.4 molar. For those of us who haven't held a pipette in a decade, give us some context. Is that a lot?
It is a staggering amount for a biological system. To give you a comparison, the concentration of glucose in the fluid outside the cell is usually around 5 millimolar. That's 0.005 molar.
Okay, so that's a huge difference.
We're talking about a concentration gradient that is wildly out of balance. You'd have almost 100 times more solute inside the cell than outside.
And nature hates imbalance.
Nature abhors it. If you have that much solute, that much sugar inside the cell, the laws of osmosis take over. Water from outside is going to rush in to try and dilute it. The osmotic pressure would be huge.
They would just flood the cell.
Leninger calculates it would be so great it would rupture the cell membrane.
So the cell swells up and...
It swells, the membrane stretches past its limit, and pop. Lysis. Cell death. Storing free glucose is essentially a death sentence because it attracts too much water. It turns the cell into a water balloon that's been overfilled.
Okay, so that's a very compelling reason not to do it. So the solution is polymerization.
Effectively, yes. Use polymerization to hide the sugar from the water. You take those thousands and thousands of individual glucose molecules, which are all osmotically active, and you link them together into one single massive particle.
The glycogen granule.
Right. By tying them all together, you drop the concentration from 0.4 molar down to about 0.01 micromolar.
Wow. That is orders of magnitude difference.
It renders the glucose osmotically invisible. You have the same amount of fuel, the same potential energy, but to the water outside, it looks like there's almost nothing there. It is a brilliant workaround to a fundamental physical chemistry problem.
That's the why. So let's talk about the what. What does this thing actually look like? Because when I was reading the description in Leninger, I wasn't picturing a long string like a spaghetti noodle. I was picturing a sphere.
That's right. It forms a granule, specifically what they call a beta granule. If you look at it under an electron microscope, it just looks like a dense, dark dot in the cytoplasm. In muscle tissue, these granules are spherical and about 21 nanometers across.
But Harper points out something really interesting here about the difference between where these granules live, the muscle versus the liver. They arrange themselves differently, don't they?
They do, and it is our first major clue that these tissues have very different personalities, very different jobs.
Okay.
In the muscle, the beta granules tend to hang out alone. They're solitary. But in the liver, they cluster, they group together like grapes to form these massive complexes called alpha granules.
And these are huge, right? The book says up to 300 nanometers.
They are massive. And Harper suggests this isn't just random clumping. It almost certainly relates to the speed of release. Well, think about surface area. The smaller, separate granules in muscle have a huge surface area relative to their volume. That allows enzymes to attack them from all sides instantly.
Okay, so more surface area, faster access.
Right. Muscle needs fuel now. When you're lifting a weight or running from a threat, seconds matter. You need to dump that glucose immediately.
Whereas the liver.
The liver is the slow-release capsule. The alpha granules, because they're clumped, release glucose a bit more slowly, more sustainably over time, which matches the liver's job perfectly. It isn't trying to help you sprint. It is trying to keep your brain alive while you sleep for 8 hours.
Form follows function. I love that. Now, we have to talk about the branched nature of this thing. We called it a tree earlier, but Leninger gets really specific about the bonds. We have alpha 1 to 4 and alpha 1 to 6.
I know, it sounds like dry chemistry jargon. But stay with me, because this is literally life or death geometry.
Okay, let's unpack the geometry.
So imagine a long chain of beads. That's the linear chain. The beads are glucose, and the string holding them together is the alpha 1 to 4 bond. It makes a straight line.
Easy enough.
But every 8 to 12 beads, nature throws a curveball, a branch point. A new chain shoots off to the side. That connection is the alpha 1 to 6 bond.
Why do we need the branches? Why not just make one incredibly long coiled rope of glucose? Wouldn't that pack tighter?
It might pack a little tighter, but it would kill you. The answer, again, is speed. Think about how we get energy out of this structure. We have enzymes that come and nibble glucose off the ends of the chains.
The ends, okay.
If you have one long rope, you have two ends, head and a tail. So you can only have two enzymes working at once.
That's a bottleneck.
It is a massive bottleneck. But if you branch that string, if you branch it every 10 units, and then branch those branches, suddenly you don't have two ends, you have thousands. A mature glycogen granule has thousands of these non-reducing ends all over its surface.
So when the adrenaline hits, when the saber-toothed tiger jumps out of the bushes, you don't have two enzymes working. You have an army.
You have a swarm.
Yeah.
Thousands of enzymes land on those tips simultaneously. It allows for an explosive, massive release of glucose. Without branching, our metabolic response would be way too sluggish. We'd be eaten before we could even warm up our muscles.
So our survival literally depends on that alpha 1 to 6 bond. That is wild.
Yep.
But there is a mystery at the very center of this granule, isn't there? We talk about adding beads to the string, but how do you start the string in the first place?
The chicken and egg problem of biochemistry. The main enzyme that builds glycogen, it's called glycogen synthase, is surprisingly incompetent in one way.
Okay.
I cannot start from scratch. I can only add to a chain that already exists. It needs a primer of at least 4 glucose units.
So if you completely starve yourself and burn all your glycogen, how do you ever build it back? There's nothing to add to.
You need a seed, a primer. And that seed is a protein called glycogen. It is buried deep in the center of every single glycogen granule in your body.
It is a protein. I thought we were talking about carbohydrates.
We are. But the core, the very heart of it, is a protein. And glycogen is fascinating because it acts as both the scaffold and the enzyme. It grabs the first glucose molecule and attaches it to itself.
It paints itself with sugar.
Literally. It has a specific tyrosine residue, TR194RAC, on its own structure, and it covalently links the glucose to it. Then it adds another and another until it has a little chain of about 8 glucoses.
And once that little chain is there,
then the big enzyme, glycogen synthase, looks at it and says, okay, I can work with this, and it takes over to build the rest of the massive particle, which can have, up to 55,000 glucose residues.
Wow. But at the heart of every sugar reserve is this tiny protein seed.
It's almost poetic, isn't it?
It really is. Okay, so we've built this massive branched water-neutral energy savings account. Now let's talk about making a withdrawal. This is part 2 of our deep dive, mobilization.
Or as the biochemists call it, glycogenolysis, the lysis of glycogen.
Now, Leninger makes a huge deal about a distinction here that I initially thought was just pedantic. He says, we don't do hydrolysis, we do phosphorylysis.
It sounds like splitting hairs, right? Hydro, phospho, what's the difference?
It does. To a layperson, you're just chopping sugar off. Why does it matter how you chop it?
It matters because of the energy bill. Think about digestion in your stomach. When you eat starch bread, pasta, your body uses hydrolysis. It uses a water molecule, hydro, to split the bonds. The result is plain free glucose.
Which is what enters the blood.
Right. But inside the cell, we don't use water, we use inorganic phosphate. It is like a molecular wedge. The enzyme glycogen phosphorylase jams a phosphate group into the bond to pop the glucose off.
So you don't get free glucose, you get what?
You get glucose 1-phosphate. It comes off the line already carrying a phosphate group. It's precharged.
And that's good.
It is fantastic. It is a major energy hack. Remember, the first step of burning glucose in glycolysis, the hexokinase step, usually requires you to spend an ATP molecule to attach a phosphate.
Right, you have to invest energy to get energy, the energy investment phase.
Exactly. But here, because we used phosphorylysis, the glucose is already phosphorylated. We effectively skip that first cost. We've got the energy of the bond itself to pay for the phosphate attachment.
So it is like getting a discount on your fuel.
Precisely. It preserves the energy potential. If we used water, we'd waste that bond energy as heat. By using phosphate, we trap it. It's incredibly efficient.
Smart. But this enzyme phosphorylase, it is the one you called the Pac-Man earlier. It eats down the chain, but it is not perfect, is it? It has a flaw.
It's a big enzyme. It's bulky. And it chews down the nice straight alpha-1,4 chains beautifully.
Yeah.
But when it gets close to one of those alpha-1,6 branch points you talked about, it gets stuck. It can't fit into the tight corner where the branch connects.
Leninger says it stops 4 residues away.
Right. It chews along until there's a little stub of 4 glucoses left on the branch, and then it stalls. It leaves what's called a limit dextrin.
And if we didn't have a way to handle that?
All the glucose deep in the granule would be trapped behind that branch. We'd lose access to most of our fuel.
Enter the debranching enzyme.
The Swiss Army knife of glycogen metabolism.
Yeah.
I love this enzyme because it has 2 completely different tools built into one protein. It's bifunctional.
Okay, walk us through the maneuver.
So we have this little nub of 4 glucoses sticking out at a branch. The debranching enzyme comes in. First, it uses its transferase activity. It acts like a little construction crane. It grabs 3 of those 4 glucoses, rips them off as a block, and pastes them onto the end of a nearby straight chain.
So it just moves the road, it extends the main highway.
Exactly. Just straightens out the path, leaving just one single glucose molecule behind, all alone at the branch point.
And that's where the second tool comes in.
The glucosidase activity. This is the only part of the process that actually uses water, hydrolysis. It just snips that last lonely branch glucose off.
And Harper highlights a really specific detail here. That single snipped glucose, is it phosphorylated?
It is not. That is the one and only time in this entire process that we produce free, uncharged glucose.
So about 90% of the glucose comes off as high-energy glucose 1-phosphate, and maybe 10%, the branch points, comes off as plain, boring glucose that we then have to spend an ATP on to use.
Correct. It's a minor inefficiency, but it is the price we pay for having that rapidly accessible branched structure.
Okay, so now we have a big pool of glucose 1-phosphate, but that isn't quite ready for the main engine, is it?
No. The main engine glycolysis wants glucose 6-phosphate. The phosphate has to be on carbon 6, not carbon 1.
So we need a traffic cop, an enzyme called phosphoglucomutase.
Right. It basically plays a little shell game. It has a serine residue with a phosphate on it. It gives its phosphate to carbon 6, takes the phosphate from carbon 1, and the net result is you've just moved the phosphate.
And once we have glucose 6-phosphate, or G6P, that's the golden ticket?
That is the metabolic crossroad. From G6P, you can go anywhere. You can send it down glycolysis to make ATP. You can shuttle it into the pentose phosphate pathway to make DNA components. Or, and this is a huge or, if you are the liver, you can ship it out to the rest of the body.
Which brings us to a really crucial distinction. We've been talking about the body generally, but Harper makes it very, very clear that when it comes to glycogen, we are really telling a tale of 2 tissues.
The liver and the muscle.
They use the same molecule, but philosophically they are complete opposites.
Total opposites.
In my notes, I labeled them. The liver is the altruist, and the muscle is the miser.
Last, that is a perfect characterization. I am absolutely stealing that.
Let's start with the altruist, the liver. What makes it so generous?
Well, the liver's biological mandate is to serve the collective. Its prime directive is to maintain blood glucose homeostasis for the tissues that can't store their own fuel, specifically the brain and red blood cells.
They're completely dependent.
Completely. When you are fasting or sleeping, your brain is still chewing through huge amounts of sugar. The liver is the organ that provides it.
But to do that, it needs a special piece of machinery that the muscle doesn't have.
The magic enzyme. It's called glucose 6-phosphatase.
This is the exit door.
This is the only exit door. Remember, we said G6P is trapped. That phosphate group acts like an anchor. Charged molecules cannot cross the cell membrane easily. If you want to release sugar into the blood, you have to rip that phosphate off.
And only the liver has the scissors to do that.
The liver and, to a lesser extent, the kidneys, but mainly the liver. And Harper goes into this really cool detail about where this enzyme is hiding. It isn't just floating around in the soup of the cell.
No, it is sequestered. It's hidden away.
It is embedded in the membrane of the endoplasmic reticulum, the ER. And the active part of the enzyme faces inside the ER lumen.
Why hide it away? Why not just have it in the cytosol with everything else?
Think about it. If glucose 6-phosphatase were floating around freely, it would constantly be stripping the phosphate off the G6P that the liver is trying to use for its own energy needs, for its own glycolysis.
It would be sabotage, a futile cycle.
Exactly. It would short-circuit the liver's own metabolism. By compartmentalizing it, the liver creates a system of control. There are specific transporters that shuttle G6P into the ER, the enzyme clips the tag, and then other transporters ship the free glucose out into the bloodstream. It's a dedicated export pathway.
So the liver works hard to feed the brain. Now let's contrast that with the muscle, the miser.
Muscle is incredibly selfish. Harper states it clearly and bluntly. Muscle glycogen is for muscle use only, period.
Because it lacks that exit door.
It completely lacks glucose 6-phosphatase. It physically cannot remove the phosphate anchor. So once glucose enters a muscle cell and gets phosphorylated by hexokinase, it is like checking into the Hotel California.
You can check out anytime you like.
But you can never leave.
I found it. I keep it.
Precisely. Even if your brain is starving, even if you are hypoglycemic and passing out, your muscles will sit on their glycogen reserves and not share a single molecule.
That seems ruthless.
It is, but it's survival. If you need to run from a bear, you don't want your leg muscles to have given away all their emergency fuel to the liver 5 minutes ago. Muscle protects its own fuel supply for acute emergencies.
So the energy dynamics are completely different. Liver glycogen vanishes during fasting.
And muscle glycogen vanishes during intense exercise.
But there is a connection between them, though, isn't there? A way they cooperate, the Cori cycle.
Yes, the peace treaty between the miser and the altruist. When muscle burns glucose really hard, like during a sprint, it does it anaerobically, without oxygen. This produces lactate, what we used to call lactic acid, as a waste product.
Which causes the burn we feel.
Right. The muscle can't reuse that lactate very well, so it just dumps it into the blood. The liver, being the altruist again, picks up that lactate. It spends its own energy through gluconeogenesis to convert that lactate back into fresh glucose, and then it ships that glucose back out for the muscle to use again.
The liver is basically doing the muscle's laundry.
And paying for the detergent. It's a very expensive process for the liver, but it allows the muscle to keep working at high intensity for longer than it otherwise could.
Okay, so we have the machinery and we have the players. But how does the body know what to do? This is part 5, the control room.
This is my favorite part, the regulation. Because it doesn't just a light switch. It isn't on or off. It is more like a nuclear launch sequence. It's an intricate, multi-layered system of control.
Leninger uses the metaphor of the cascade, the waterfall.
The waterfall. This is so important to grasp. It explains the concept of amplification. You might have a tiny, tiny amount of hormone in the blood-like epinephrine, maybe 10 to the power of minus 10 molar, vanishingly small. A few molecules.
But the effect is massive.
Because that one hormone molecule hits one receptor, which activates, say, 10 adenyl cyclase enzymes inside the cell. Those 10 enzymes produce 100 molecules of cyclic AMP. Those 100 activate 1,000 protein kinases.
And on down the waterfall.
By the time you get to the bottom of the waterfall, to glycogen phosphorylase releasing glucose, you have millions of molecules moving. It turns a whisper into a scream.
From a single hormone molecule.
Exactly.
So let's meet the signal callers. We have 3 main hormones on the control board.
Glucagon, epinephrine and insulin.
Let's start with glucagon.
That's the signal for hunger. It is released by the alpha cells of the pancreas when blood sugar drops. It speaks almost exclusively to the liver. Its message is simple. Hey, the brain is running low on fuel. Open the vault.
And it doesn't talk to muscle.
Muscle doesn't even have glucagon receptors. It's deaf to that signal.
Okay, epinephrine.
Adrenaline. That's the signal for terror or action. It's the fight-or-flight hormone. It speaks to both the liver and the muscle. It screams, "We need energy everywhere right now. Don't ask questions, just burn."
And insulin.
The Fed signal, the peacemaker. It's released by the beta cells of the pancreas when blood sugar's high after a meal. It says, "We just ate a pizza. Everybody calm down, stop burning, and start packing this stuff away for later."
So the mechanism here is elegant. It uses phosphorylation, adding phosphate groups to enzymes as a master switch.
Right. When epinephrine or glucagon hits its receptor, it triggers that cascade we talked about, leading to a rise in a molecule called cyclic AMP or CANT-B. This activates the master kinase, protein kinase, APKA.
PKA is the general.
PKA is the general. It marches through the cell and starts tagging other enzymes with phosphates. It finds an enzyme called phosphorylase kinase and phosphorylates it, which activates it. That enzyme then finds glycogen phosphorylase and phosphorylates it, turning it on.
So phosphate means go for breakdown.
In this case, yes. But here is the genius part that Leninger highlights so well. Reciprocal control. You do not want to be building glycogen at the same time you are trying to burn it.
That would be a futile cycle.
You'd just be spinning your wheels and generating heat. It's incredibly wasteful.
So how does the cell prevent that?
The same general PKA that turns on the breakdown crew also goes over to the construction crew, glycogen synthase, and slaps the phosphate on it.
And what does that do to the synthase?
It turns it all off. It paralyzes it. So the single signal, epinephrine, simultaneously hits the gas on burning and the brakes on building. It coordinates the whole factory with one command. It's beautiful.
That is smart engineering, but there are exceptions. The system isn't purely top-down. Leninger and Harper both talk about allosteric override.
Right. This is the local branch manager overriding the CEO from headquarters.
Give me an example in muscle.
Okay, look at muscle. Let's say you get jumped by a predator. You haven't even had time to release adrenaline yet from your adrenal glands, but you react instantly. Your muscles fire. You consume APP in a millisecond.
And when you use ATP, you're left with AMP.
Exactly. AMP is the debris of used energy. It acts as a local distress flare inside that muscle cell. AMP binds directly to glycogen phosphorylase and forces it to pop open and start working. It doesn't wait for the hormone cascade. It bypasses the chain of command because the need is immediate.
I don't care what headquarters says. We need fuel in this specific muscle cell right now.
Precisely. And calcium does the same thing. Calcium's the ion that triggers muscle contraction. That same wave of calcium that tells the fibers to contract also leaks over and hits phosphorylase kinase, activating breakdown. It synchronizes the fuel supply with the movement perfectly.
It makes so much sense. And the liver, it has an override too, right?
For a different job. It has the glucose sensor. This is a negative feedback loop. If you eat a big meal and blood sugar is already high, glucose flows into the liver cells via the GLUT2 transporter. That free glucose binds directly to the active breakdown enzyme phosphorylase and essentially changes its shape to say, stop, we have enough.
The tank is full, stop pumping.
It makes it a better target for a phosphatase, an enzyme called PP1, which comes along, snips off the activating phosphate, and shuts it down. So even if you have some adrenaline floating around from stress, if your blood sugar is super high, the liver won't dump more fuel into the fire. It's a failsafe.
Okay. We can't leave the control room without talking about the fed state. Synthesis. We talked about how breakdown works, but Leninger emphasizes that synthesis is not just the reverse of breakdown.
This is a fundamental rule of metabolism and control. If synthesis were just the reverse pathway, you couldn't regulate them separately and efficiently. They are separate roads with different enzymes and different regulation.
And to build glycogen, we first need to activate the glucose. We need to attach a tag to it.
UDP glucose, uridine diphosphate glucose.
Leninger mentions Louis Loire here, a Nobel Prize winner.
The Argentine biochemist, yeah. He figured this out. You can't just stick glucose onto a growing chain. Energetically, it is an uphill battle. It's not favorable. You need to give it a push.
But the UDP is the push.
Exactly. The cell first reacts glucose with UTP, another high-energy nucleotide. Attaching the glucose to the UDP releases a pyrophosphate, which is then immediately broken down, and that whole process releases a ton of energy. It is the sunk cost that makes the final addition of glucose to the chain irreversible and favorable.
So insulin is the hormone that triggers this storage process. How does it undo all that fight or flight signaling?
Insulin is the great dephosphorylator. It activates enzymes, mainly protein phosphatase 1 or PP1, that go around snipping off all those phosphate groups that epinephrine and pKa added.
It resets the board.
It calms the breakdown enzymes by dephosphorylating them, turning them off, and wakes up the synthesis enzymes by dephosphorylating them, turning them on. Leninger also mentions a specific bully enzyme called GSK3 glycogen synthase kinase 3.
What does the bully do?
Its sole job is to keep glycogen synthase turned off by constantly adding phosphates to it. It just sits on it. Insulin comes in and, through its own signaling cascade, phosphorylates and inactivates GSK3. It essentially handcuffs the bully.
So it inhibits the inhibitor.
Right. With the bully gone, PP1 can come in, clean up the phosphates, and glycogen synthase can finally get to work and store that pasta you just ate.
It is such a complex web. Everything is connected. But when those connections break, that brings us to our final section, Part 6, the clinical detective.
Glycogen storage diseases, GSDs.
Harper calls these experiments of nature. It is a slightly cold way to put it, but it's true. These rare genetic diseases prove exactly what each enzyme does by showing us the catastrophic consequences when it is gone.
And tragically, they are often devastating. Let's look at type I, von Gierke disease.
This goes right back to our ulcerous liver and that magic enzyme.
Right. These patients are born without functional glucose 6 phosphatase. They have the trap. Their liver can take up glucose, it can turn it into glycogen, it can even break that glycogen down to G6P. But it cannot release the free glucose. The exit door is locked shut.
So the liver just fills up with glycogen and G6P.
It becomes engorged. Hepatomegaly, a massive swollen liver, is a key sign. But the immediate threat is the blood sugar. Because the liver can't release glucose during fasting, the patient suffers severe, life-threatening hypoglycemia.
So if they miss a meal...
If they don't eat for 3 or 4 hours, their blood sugar crashes to lethal levels. Their brain, which is totally dependent on that glucose, has no fuel source.
And Harper mentions lactic acidosis too. Why that?
Because that trapped G6P has to go somewhere. The liver shoves it down the glycolysis pathway at a furious rate, producing massive amounts of pyruvate, which gets converted to lactate and fat. So these kids have high lactate, high lipids, and a massive liver, but they are starving on the inside.
The treatment mentioned is surprisingly low-tech, uncooked cornstarch.
It works. Cornstarch is a complex carbohydrate that digests very slowly in the gut. It acts like a continuous IV drip of glucose. If you feed the patient cornstarch every few hours, even through the night via a tube, the liver never needs to release its stores. You bypass the broken step by providing a constant external supply.
Amazing. Then there is type 2, Pompe disease. This one feels different. It isn't about blood sugar regulation, is it?
No, Pompe is a lysosomal storage disease. It is a housekeeping problem. Remember, we said most glycogen is in the cytosol, but a tiny, tiny fraction gets swept up into lysosomes, the cell's garbage disposals, just as part of normal cellular turnover.
And to break it down in the lysosome, you need a specific enzyme that works in that acidic environment.
Acid maltase, or lysosomal alpha-glucosidase. In Pompe disease, this enzyme is missing, so that tiny fraction of glycogen goes into the lysosome, but it never comes out. It just accumulates bit by bit.
It is like a trash compactor that never runs. The bags just keep piling up.
Exactly. Over time, the lysosomes swell up until they are gigantic. They eventually take up so much space that they physically disrupt and destroy the structural integrity of the muscle fibers.
And this hits the heart the hardest.
Cardiomegaly. The heart muscle becomes so thickened, stiff, and weak from these glycogen-stuffed lysosomes that it fails. In the infantile form, it is often fatal very early in life. It's a stark reminder that glycogen metabolism isn't just about fuel. It is about cellular hygiene.
Let's talk about type V, myocardial disease. This is the muscle one, the miser.
Right, this is a defect in muscle phosphorylase. The Pac-Man enzyme in the muscle is broken.
So the muscle is full of glycogen.
Stuffed with it. It has a full tank of high-octane fuel, but it's locked away. The key is missing.
The symptoms are exercise intolerance, cramps. But there is a classic diagnostic sign Harper mentions that I found fascinating, the second wind.
It is a distinct clinical phenomenon. A patient with McArdle's starts to exercise, say, jogging or climbing stairs. Immediately, they are in agony. Their muscles cramp up because they cannot access the glycogen fuel they need for that initial burst of anaerobic activity.
They hit a wall instantly.
But if they stop, rest for a minute, and then start again gently, they suddenly feel better. They get their second wind and can keep going, sometimes for a long time.
Why? What changed in that minute?
The blood flow caught up. In the first few minutes of exercise, you rely on stored muscle glycogen. But after about 10 minutes, your heart rate is up, your arteries have dilated, and you are delivering blood-borne fuels-free fatty acids from your fat stores and glucose from the liver to the muscle.
So they switch fuels from their own broken supply to the public supply.
Exactly. They survive the glycolytic failure and move into aerobic metabolism using fuels delivered by the blood. That second wind is the moment the fuel source switches. It proves perfectly that muscle glycogen is primarily for that initial anaerobic burst.
That is such a clear illustration of the physiology. Finally, just a brief mention of type IV Anderson disease, the branching enzyme.
The branching enzyme defect. This is truly tragic. If you lack the branching enzyme, you still build glucose chains, but they are long and straight, with very few branches.
Basically amylopectin, like plant starch?
Yes. And the human body does not like starch growing inside its liver cells. The immune system recognizes these long, straight, insoluble chains as foreign bodies.
So the body attacks itself.
It launches an immune attack on the liver. It leads to severe scarring, cirrhosis, and ultimately liver failure in childhood. It shows that the shape of the molecule, the branching, is just as important as the chemistry. If it isn't branched, it isn't just inefficient. It is toxic.
So we've unpacked the structure, the regulation, the pathology. When you step back and look at this entire system, what is the big takeaway?
For me, the big takeaway is that glycogen is insurance.
Insurance against what?
Against the unpredictable nature of the world. We live in a reality where fuel isn't always available and threats can appear at any moment. Glycogen buys us time. It buys the brain time between meals so we don't faint. It buys the muscle time to escape a threat before the heart can catch up.
And the elegance of the system. The way the liver sacrifices for the brain while the muscle hoards for the fight. The way synthesis and breakdown are reciprocally controlled so we never waste energy.
It is the definition of metabolic efficiency. It is a system honed by millions of years of starve or die, fight or be eaten pressure.
Here is a thought to leave our listener with. We store way, way more fat than glycogen. A typical 70-kilogram person has maybe 15 kilograms of fat enough energy to walk to the next state, but we only have about half a kilo of glycogen, maybe 2,000 calories worth.
It's a tiny tank in comparison.
So why bother? Why keep this complex, heavy, waterlogged glycogen system at all? Why not just run on the much more efficient fat reserves?
That is the provocative question, and the answer is oxygen.
Explain.
Burning fat is a slow and steady process. It absolutely requires oxygen. It is purely aerobic, but glycogen can be burned anaerobically via glycolysis. It does not need oxygen to generate ATP in the short term.
It is the turbo boost, the nitro.
It is the only fuel that can save you when you have to run so fast that you can't breathe. If a lion is chasing you, you don't have time to mobilize fat from your adipose tissue and slowly burn it. You need energy faster than your lungs can deliver oxygen. Glycogen provides that.
So fat keeps you alive during a famine, but glycogen keeps you alive during a fight.
Precisely. It is the metabolic price we pay for being able to survive acute stress. It's our emergency power supply.
That puts it all in perspective. I'd encourage everyone to open up Leninger to the chapter on glycogen metabolism or Harper to chapter 18. Look at those cascade diagrams. It is one thing to hear about the waterfall. It is another to see the arrows connecting pKa to phosphorylase.
It really connects the molecular dots to the physiological reality.
Thanks for diving deep with us today.
My pleasure.
See you next time.